Perfect. Looks great.
Can you see my presentation?
Yeah, looks fantastic.
Okay. Good afternoon, everyone. Thank you for joining me here today. So my name is Anu Jose. I am a PhD candidate at Department of Biochemistry and Molecular Biology at Dalhousie Medicine, New Brunswick, and Dalhousie University. So today I'm going to talk about the role of a phosphatase called lipid phosphatase 3 in cardiac lipid and mitochondrial homeostasis, and how it can safeguard heart against obesity-induced cardiac dysfunction. Okay. And then, we all know that the heart is one of the metabolically active organs in the body and requires a continuous supply of ATP to sustain contractile function. And cardiomyocytes rely heavily on mitochondrial oxidative metabolism, with mitochondria generate almost 90% of the ATP required for cardiac contraction. In fact, heart actually turns over almost entire pool of ATP every few seconds. And so it highlights the extraordinary demand for the heart to maintain normal cardiac function. Therefore, it is very important that to know about how we can a mechanism relating to protect heart against any kind of stress is really, really important. So obesity is like a key contributor to metabolic syndrome. It actually disrupts metabolism and increases the risk of cardiovascular diseases. So, like for example, during obesity, what happens is this adipose tissue expands and becomes inflamed, and it leads to lipid spillover to other tissues, including the heart, and leading to different metabolic dysfunction. Uh, and it also leads to different types of cardiac conditions, like, for example, cardiomyopathy, which is the left ventricular enlargement in the absence of coronary artery disease, which is also a common cause of heart failure. So as mentioned, there are different types of lipid spillover happens during adiposity because of the adipocyte fibrosis. So one such lipid that actually increased during obesity is lysophosphoric acid, or LPA, and it has been, so like from this graph you can see here that this is from the mouse, that the systemic LPA levels goes up with obesity in mouse. It has also been shown in humans as well, also in other more metabolically related issues. So also it has been also shown that this increased LPA levels actually contribute to mitochondrial dysfunction, and a mitochondrial dysfunction leading to different conditions. So it is actually very important to study or look into the why this is getting increased during obesity, and can we actually study more about by reducing the levels and help the metabolically active organs or not. Similar to, so as mentioned, elevated LPA is associated with different conditions, including cardiac remodeling. This is one of the study that I was able to find that actually shows that during myocardial infarction, where increased level of LPA actually increased cardiomyocyte size, which you can see here, and it is also associated with increased troponin, which is a marker for elevated cardiac injury. Elevated cardiac injury. Similar to that, another study from a pediatric dilated cardiomyopathy patients, it has shown that in using clinical lipidomic studies that circulating lysophosphoric acid also correlated with cardiac remodeling, such as left ventricular ejection fraction and fractional shortening, which are the marker for cardiac function. And it is related to those cardiac remodeling from these patients. So therefore, mechanisms that regulate this LPA levels may be something, may be critical for maintaining cardiac. It is important to study that kind of pathways for maintaining cardiac homeostasis. So what is actually this LPA and what is actually the role of LPP3 in this pathway is, so to understand that, we have to look at the basic pathway which is called autotaxin LPA LPP3 signaling axis, which is an important signaling pathway. So lysophosphatidic acid actually produced from lysophosphatidylcholine choline by an enzyme called autotaxin in the extracellular space, and this LPA acts on different G protein coupled receptor called LPA. There are 6 identified G protein coupled receptors, which is from LPA receptor 1 to 6, and which is on the cell membrane. And activation of these receptors actually triggers several downstream pathways, which are very important for the cell survival and growth, including Rho signaling, PLC signaling, MAPK signaling, PI3K, and adenylate cyclase signaling. So what is the role of LPP3 here is to control this LPA signaling. LPP3 is actually known to dephosphorylate this LPA, and it converts to monoacyl glycerol and thereby terminating LPA signaling. So basically, LPP3 controls LPA signaling by degrading LPA. So that is what my research primarily focuses on. So to further understand whether LPP3 is involved in any metabolic diseases, because it is a major brake on LPA signaling. So we asked whether it is related to any metabolic disease or not. So to study that, so what we actually done is we compared human atrial appendages samples which is collected through a clinical trial study called OPOS, known as Impact of Obesity on Post-Operative Outcomes Following Cardiac Surgery. So it was a partnership with St. John Regional Hospital here at New Brunswick. So we had samples that were patients who were non-diabetic and also diabetic. So as you can see here, that there was a clear reduction in LPP3 protein expression in the diabetic patients. We also confirmed this in STZ-induced diabetic rats, where we saw a similar kind of results where LPP3 levels were significantly lower in diabetic controls compared to diabetic rats compared to the normal rats. So across both human and experimental models, we saw clear change in LPP3 protein pattern when reduction. So we thought that, okay, maybe it has something to do with not clearing LPA levels systematically or maybe locally in the cardiac tissue itself. So our next question was whether enhancing LPP3 expression mitigate cardiometabolic dysfunction by altering the LPA levels or not. So to study that, so this study is already published in AJP Heart and Physiology, so circulatory physiology. So to further study that, what we have used is I had a mouse model where we had overexpressed LPP3 just in the heart, and then we fed this mice low fat and high fat diet to induce obesity for over 22 to 24 weeks. And then we assessed different cardiometabolic outcomes, including plasma LPA, cardiac function, adiposity, mitochondrial respiration, and the results were really interesting. So particularly in female mice with LPP3 overexpression, it actually reduced the plasma LPA levels as well as LPA signaling, and also insulin sensitivity was preserved. Also, these mice had cardiac function were protected, also showed an enhanced pyruvate-linked respiration following high-fat diet feeding, which was pretty cool, which we did not expect. But the another interesting thing was male LPP3 overexpressing mice did not show any improved protective effect from LPP3 overexpression. So either way, we think that it demonstrates that enhancing LPP3 provides, even though it's a sex-specific cardiometabolic function, it is actually showing, okay, LPP3 might be involved in this particular metabolic pathway. So so all of these findings led us to the next fundamental question of this research. So what is exactly the role of this pathway, LPA-LPP3 pathway in cardiomyocytes to influence the cardiac metabolic
Uh,
remodeling? So, and we think LPP3 is involved under conditions such as mitochondrial function and lipid homeostasis, because these are the two important metabolic dysfunction that usually happens with obesity and other conditions like type 2 diabetes, etc. So I wanted to look at the how this pathway is involved in the mitochondrial function, cardiac mitochondrial function, and lipid droplet homeostasis. So first, first I looked at the mitochondrial function. So my first question was to determine whether LPA directly affects mitochondrial function or not. So I used neonatal rat cardiomyocytes, isolated neonatal rat cardiomyocytes from 1 to 2 days old rat pups, and treated with 10 micromolar LPA for 18 hours. It's a pathophysiological concentration to stimulate the LPA signaling, and measured oxygen consumption. We used a substrate uncoupler protocol with malate, pyruvate, ADP, rotenone, succinate, and AMA to assess different states of mitochondrial respiration. And you can see here, compared with the BSA controls, when the cardiomyocytes treated with LPA actually showed significantly reduced ADP stimulated respiration, as well as when with succinate linked respiration, indicating the complex linked respiration, which was also reduced, which reflects the mitochondrial oxidation phosphorylation capacity is reduced. So overall, these results indicate that LPA diminishes mitochondrial respiration in cardiomyocytes. So next, we wanted to see what is the role of LPP3 in cardiomyocytes itself. Earlier in our in vivo model, we saw with LPP3 overexpression, pyruvate linked respiration was going up. So next, we wanted to see what happens if LPP3 is not around when we remove LPP3 from the cardiomyocytes. So what I actually did was I did a knockdown in NRCMs, neonatal rat cardiomyocytes, and I measured oxygen consumption rate. And as you can see here from the purple graph, that with LPP3 knockdown, it led to a strong decrease in mitochondrial oxygen consumption, indicating impaired mitochondrial respiration. And then I examined proteins involved in mitochondrial dynamics, such as OPA1 and mitofusin-1. Both are involved in mitochondrial fusion. So in the absence of LPP3, you can see here that the proteins involved in mitochondrial fusion is reduced. And with LPA, the effect is further exacerbated. So suggesting that LPP3 is important for mitochondrial respiration and is involved in mitochondrial quality control mechanism. Further studies are required to further understand what is happening here. So that will be like my future studies. And then after that, I also overexpressed LPP3 in the neonatal cardiomyocytes itself. And so here you can see here when I overexpressed so I used two different virus here, which is a wild type LPP3. So it overexpresses the increases the LPP3, and there is a mutant LPP3 as well, which actually it's like the catalytic, it's like a catalytically inactive. So the activity of the LPP3 is mutated here by at the position S19, certain 197T position. So you can see here with wild type overexpression, the mitochondrial respiration is increased, while the inactive mutant did not affect the mitochondrial respiration. So meaning that the catalytically activity of LPP3 is required to change the respiration phenotype in cardiomyocytes. So after that, because we saw different changes in mitochondrial respiration, we also looked at the mitochondrial abundance and the biogenesis markers. So for example, here you can see here with active LPP3 being overexpressed, the TFAM protein levels, which is an indicator of biogenesis, is increased, and as well as the citrates in this. It's a marker, mitochondrial abundance marker. So for both of them were increased with LPP3 overexpression in neonatal cardiomyocytes. So which was something similar to our what we have observed with our in vivo model, where we saw TFAM mRNA levels were increased in our cardiac specific LPP3 in from the heart from the cardiac specific LPP3 overexpressing mice, as well as the citrates in this activity. So suggesting that whether in vivo or in vitro, when we try to overexpress LPP3, mitochondrial biogenesis and abundance increases, and further studies are also required to understand how this is happening. Then my next part of my study is to look at the lipid droplet homeostasis. So what we actually did was I actually isolated adult mouse cardiomyocytes from the LPP3 overexpressing mice, and using untargeted lipidomics, we measured triglyceride levels, because triglyceride levels are an indicator of the lipid droplet accumulation in the cardiomyocytes. So following high-fat diet, and in female mice, we actually observed this cardiac specific LPP3 overexpression significantly reduced this tag levels, which was actually supporting to the preserved cardiac function and mitochondrial respiration. But we did not observe this in the male mice, and then after that, I looked at lipolysis marker, which is phospho-HSL serine 660. So you can see here the phospho-HSL serine 660 was upregulated. So it indicates that phosphorylation of HSL at serine 660 indicates increased lipolysis, which was further supporting our triglyceride levels in the mice. So together, these data shows that cardiac LPP3 overexpression reduces the TG accumulation by enhancing the lipolysis in the cardiomyocytes. And then I further wanted to look at the lipid droplet accumulation in cardiomyocytes when LPP3 is not present. So again, what I did was I actually knocked down LPP3 in neonatal rat cardiomyocytes, and then in the presence and absence of LPA. So you can see here that as shown in this images, LPA-induced lipid droplet, there are high lipid droplet accumulation with LPA, and then in the absence of LPP3, it actually further exacerbated. So suggesting that loss of LPP3 drives LPA excessive, the excessive LPA-induced lipid droplet accumulation. We also looked at the HSL phosphorylation again to see whether it is linked to the lipolysis activity or lipolysis function. And then, as you can see here, the LPA did reduce the HSL phosphorylation, indicating reduced lipolysis, and without LPP3, it was further exacerbated. So that means when LPP3, so in cardiomyocytes, LPP3 is involved in lipolysis pathway to change the lipid droplet homeostasis. And when LPP3 is not around, the lipolysis is inhibited. And further to so in my coming slides, I'll be explaining, okay, which mechanism is actually leading to this kind of lipid droplet accumulation in cardiomyocytes. So to further understand this more, we used to determine whether this is consistent in a human cardiomyocyte model. So to address that question, I used a stable LPP3 knockout AC16 cells, and which has a complete loss of LPP3, and which you can see here from the Western blot, the confirmation of LPP3 loss. So similar to what I've observed in the neonatal cardiomyocytes, so loss of LPP3 markedly exacerbated the LP-induced lipid droplet accumulation. And then, so suggesting that the protective role of LPP3 limited when is limiting LPA-driven lipid storage in cardiomyocytes. And then next, I wanted to see whether LPA-driven lipid droplet accumulation is receptor mediated or not. So I inhibited LPA receptor using a chemical called key 16425, which is a pharmacological antagonist of LPA receptor, particularly LPA1 and LPA receptor 1 and 3. And interestingly, I saw that when I treated with LPA, similar to what I observed with other NRCMs and the AC16 cells, LPA did increase the lipid droplet accumulation, and then LPP3 knockdown further increased that. So when I inhibited LPA receptor signaling using key 16425 in both knockdown and in LPA group, uh, it actually reduced lipid droplet accumulation significantly. So it suggests that this is involved. This LPA induced lipid droplet accumulation is LPA receptor dependent. And then next, I wanted to see whether activating lipolysis, because we saw a reduced lipolysis with LPA induced LD accumulation. So I thought, okay, maybe if I activate lipolysis, can I reverse LP induced LD accumulation in cardiomyocytes or not? So for that, I used a drug called forskolin, which basically activate adenylate cyclase. So adenylate cyclase activate protein kinase A or phosphorylated protein kinase A, and then protein kinase A further phosphorylates different lipolysis related proteins. So, very interestingly, when LPA forskolin treatment actually reduced LPA-induced lipid droplet accumulation in both the presence and absence of LPP3. So this suggests that PK activation reduced LP-induced LD accumulation in RCM, and activating lipolysis could be the pathway that is involved in LPP3 linked lipolysis activation. And then my final question was whether overexpressing LPP3 levels could mimic the effect that we observed with forskolin-mediated lipolysis or not. So in AC16 cells, I overexpressed LPP3 using adenovirus and then treated with LPA to see whether it mimics the effects of forskolin or not. So as you can see here from the quantification data or from the images, there is significant reduction in the LD count per nuclei in LPP3 overexpressing cells upon LPA treatment. So together with the forskolin data, this suggests that LPP3 limits LPA-driven lipid accumulation. It might be through the PK-linked lipolysis activation. So to wrap this up, to know what is the role of LPP3 in obesity due to cardiac dysfunction. So it's basically LPP3 deficiency enhances LP-induced lipid droplet accumulation and as well as mitochondrial efficiency. And then PKA-mediated lipolysis attenuates LPA-driven lipid droplet accumulation. And then overexpression of LPP3 helps restore balance in both lipid turnover and supports mitochondrial integrity. So my future directions would be how LPP3 regulates mitochondrial quality control. So, and then also define mechanisms underlying LP-induced lipid droplet accumulation, including whether it is involved in de novo lipogenesis, lipolysis, or even lipophagy. Also, how or identify which are the LPA receptors that actually mediates this metabolic remodeling. So ultimately, these studies will define how this axis regulate cardiomyocyte metabolic remodeling and can be used in different research that are related to heart failure or any like cardiomyopathy, etc. And then that's all. And with that, I would like to thank all of my lab mates, especially my supervisor, Dr. Petra Kienesberger, and my committee members from university and all the research funding groups. Thank you.
Awesome. Thanks so much, Anu. I really appreciate that. If you have any questions, feel free to ask Anu or type it in the chat and I can read it out for you. I had one question when you were talking about the male versus female for HSL.
Mhm.
Um, are you interested in probing why there's higher phosphorylation with female versus male? Or like, is that
Yeah,
something that,
yeah. We think it might be. It's obviously it's sex differences. Something has had happened with sex during the high fat feeding. I'm pretty sure about that. Something that changed. So with the LPA during high fat diet feeding in males and females, we don't know exactly what's the reason behind that, and we might be looking at, and we think that there are researches that is recently happening that is related to extracellular vesicles. So it might be something to do that, but I honestly don't know. We may be looking at different things, but not for now. I'm particularly interested in this pathway, how this is changing, and like more like the biology of this pathway.
nice. Okay.
Yeah.
In that pathway, did you ever see any sex differences at all? Like other than
It's
that?
other than basically the lipolysis, but it has been, I think, shown depending upon, when mice is in estrous cycle, all those things are really dependent on that. So I am not really sure about it. Yeah.
That's fair. Yeah, no, that's an interesting phenotype, which would be cool
Yeah.
to probe
Yeah.
further. But awesome.
Yeah.
Okay. Any other questions for either Sean or Anu? Feel free to type them in the chat. You can also reach out to them individually if you'd like. Okay. If not, we can definitely end early. Thank you both for speaking today. Really appreciate it. Again, we're going to have a bit of a break from presentations next month, but we will have a seminar still. It'll most likely be an EDI workshop, so things you can implement into your own lab. And then in May, we have our ASBMB and PMBMB speakers presenting, so feel free to stay tuned for that. But thank you so much for everyone for coming. I appreciate it, and have a great day.
Thank you.